related to the molecular biological techniques. Nowadays, number of the person they are working on the cell line because most of the time we feel that difficulty in the animals, so most of the people they are shifted over here. So I hope that this webinar will be the very helpful and beneficial to us. So Mehanji, you have given the time at what time we have start sharp at 3 o'clock?
Definitely, sir. Sir, if Dr. YK Gupta is delayed, then I talk to Dr. Bowles that she will continue, and after that Dr. S.S. Sharma will assist.
So Dr. Mehanji, what is about YouTube? YouTube is now start. You are ready?
Yes, sir. Yes, sir. So YouTube is live now.
Okay.
YouTube is live now, sir.
I think it's a great initiative
Should
by
I start, sir?
Dr. Gupta.
Yes, sir. Sir, you are audible, sir. Dr. Sharma, you are audible, sir.
Okay. Yeah, I was just mentioning, I think it's a great initiative taken by Dr. G.D. Gupta uh, for all the students and particularly pharmacy graduates, so that I think their knowledge can be enhanced in the area of molecular biology, and particularly I think COVID pandemic have taught us actually how this molecular biology has helped all of us actually, whether in terms of detection or whether in terms of the development of vaccines and all. So that's why I think molecular biology have played a very significant role in this pandemic, and we hope in future also is going to play a great role in that one.
Thank you, sir. So I request to Dr. G.D. Gupta, sir, please allow me to start this seminar. Okay, sir. So am I audible to all speakers and our dignitaries?
Yes.
Yes, thank you.
Yes.
So
Pleasure.
very good afternoon to all of you. Myself, Dr. Siddharth Mehan, working as a associate professor, Department of Pharmacology, ISF College of Pharmacy, Moga, Punjab. First of all, I would like to welcome our Chairman Shri Parveen Garg ji, Secretary Engineer Janesh Garg, and Chairman TLF A Global School Moga, Director Professor Dr. G.D. Gupta, Vice Principal Dr. R.K. Naran, Chief Guest and Speaker Dr. Y.K. Gupta, President AIMS Bhopal and AIMS Jammu, our Guest of Honor and Speaker Dr. Mike Pauls, Scientific Advisor London, Guest of Honor and our respected speaker Dr. S.S. Sharma, Department of Pharmacology, Naipur, Mohali. On the behalf of whole ISF College of Pharmacy, I welcome you all on one-day international seminar, JOVE, Cambridge, USA, on advancement in pharmaceutical education and research, current insights into the molecular biological techniques. JOVE Cambridge, which is basically a journal of visualized experiment, is the leading producer and publisher of video resources for science education and research. With thousands of videos published across several disciplines in science, medicine, and engineering, the JOVE library provides clear detailed illustrations of a vast range of scientific technique and concepts. I also welcome to our audience who is now live on YouTube. So now I request to our first guest, Dr. Mike Pauls, for their lecture. Before starting this lecture, I want to introduce Dr. Mike Pauls. Dr. Mike obtained a PhD in cell biology from Urchet University in 2012, where she was involved in teaching and research on lysosome biogenesis. She did a postdoc at the University of Queensland, where she worked on zebrafish muscle development and then moved to London to work as an editor for F1000 Research before JOVE in 2017. I welcome you, Dr. Pauls, on the behalf of ISF College of Pharmacy. Please.
Thanks. Thanks everyone for having us here today. Let me share with you our PowerPoint for today. Here we are. So I hope this is visible to everyone. So today I would love to talk to you about how JOVE can help you learn about science and also fast track your experiment using our video database. So thank you so much for that kind introduction. So a little bit about me. I am JOVE's scientific advisor. So it's my pleasure today to introduce JOVE and explain what exactly it is and what we do. I'll also show you how you can set up a free JOVE account to access some of our content remotely, so you can have a look for yourselves if any of this today sounds of interest to you. Then I'm going to introduce JOVE's research and education products with a special focus on molecular biology and pharmaceutical sciences. I'll show you how to navigate the JOVE website and also how you can create playlists that match your research interests or that can match your course topics. I'll also show you, if you are a faculty member, where you can find help creating, for example, a playlist that exactly matches your course topics, as well as help with embedding videos in your course programs online. We'll leave some time for questions at the end as well, so do save up any questions you might have. So what is JOVE exactly? So JOVE stands for the Journal of Visualized Experiments, and we create scientific videos that make teaching, learning, and practicing science more efficient, effective, and engaging. We got started back in 2006 when our CEO and co-founder Moshe Pritzker was doing his PhD at Princeton University. Now, Moshe, like a lot of young researchers, was kickstarting his own projects by trying to replicate an experiment that he read about in a science journal. Now, in Moshe's case, this had to do with the differentiation of a certain type of stem cell, but he had nobody in the lab who could help him with this experiment, and the experiment kept on not working. Now, frustrating as this was, the solution for Moshe was to send him all the way from New Jersey in America to Edinburgh, Scotland, to learn how to do this experiment in the original author's lab. This was an awesome experience for him, but it also took 2 months of traveling and cost around $10,000 just to learn this one experiment. Now, Moshe realized that this was not a great solution for everybody. So when he finished up his PhD, he decided to start the world's first video science journal to help other researchers learn new techniques that people have published from elsewhere in the world. Now, Moshe wasn't the only one who struggles with reproducibility. In fact, only 10 to 40% of published scientific articles are reproducible. There's a lot of reasons for this. For example, there's a lot of pressure to publish quickly to get your next job or your next round of funding. In some cases, not enough of the data is shared to sufficiently reproduce something, or there's by far the most common reason why other scientists cannot reproduce each other's work is unclear written protocols. There's word limits to certain articles, and it also means that people abbreviate to the point where the protocol is unclear. Now, this is where JOVE tries to solve the problem. So JOVE has created a video database with articles on research topics as well as education topics to help people learn new techniques and help students learn to work in the lab safely. So in JOVE Research, you find our video science journal, which is fully peer-reviewed and indexed in PubMed. And we also have a video encyclopedia that helps you learn advanced research techniques more efficiently and with greater reproducibility. Then we also have a JOVE education database, and this is aimed to help teachers help their students learn to work in the lab. So this is really to set up your first experiments, learn how to use certain pieces of equipment in the lab, and then carry on learning new techniques throughout the different scientific disciplines. Now, why is visualizing science so helpful? If you're trying to replicate a protocol from a text article alone, this can be really daunting, especially if you haven't worked with this type of model organism before, or if you're trying some technique from a field you're unfamiliar with. So take this example here. This is a piece of text from one of our abstracts, and it describes how to line up C. elegans worms for an imaging experiment. Worked with these worms before, or you're not sure how to image these animals, this text can be very unhelpful, and you might question what the experiment will look like when you're performing it yourself. Now, even by adding as little as a still image from this experiment, you get a much clearer picture what the authors mean when they say line up the animals in a certain way. Now, by having a video that can show you exactly how every step of the experiment will look when you're performing it, it makes it so much easier to do it exactly like that. Now, JOVE Today is a large video database. We have over 13,000 videos in our library, and on average, we publish around 1,200 new videos every year. We already have more than 1,000 institutes worldwide that subscribe to our services and well over 8 million unique users. Now, all of the videos I'm going to be talking about are accessible to you worldwide 24/7. And for our educational videos, we're constantly adding new languages and subtitle translations, which makes it easier for students to follow the video in a language that they're comfortable with. We also have well over 10,000 downloadable protocols, and our videos are compatible with most digital learning platforms. So this means if you want to use the videos in your courses, you can fully embed them into your learning management systems, such as Blackboard, Moodle, or Canvas. Now, accessing the JOVE videos is pretty straightforward. When your university subscribes to us and you're on your university campus, we use IP recognition. So this means if you go to jove.com, the videos within your subscription will play automatically. If you're off campus, we've got a couple of different ways to verify the access. For example, you can set up a JOVE account with your institutional email address, and this will link you to your university subscription. We can also work with single sign-ons such as Open Athens or Shibboleth, or set up an easy proxy for your school. By far the easiest way to share the videos with off-campus students is by embedding the videos into your course pages on your learning management system. This ensures that only you as the instructor need to set up a JOVE account, and your students can simply access the videos through your course pages and do not need to set up a separate JOVE account. Now, accessing the videos through a JOVE account has some additional benefits, and this is just to quickly show you how easy it is to set one up. So if you visit jove.com and click on the Sign In button, you can select Create an Account. Here, you use your institutional email address to set up a new account, and it is important to let us know if you are a student, a librarian, or a faculty member or teacher, as we can optimize your account for your role type. You will be sent one verification email, and once verified, your account is active and you can immediately start to use it. With your JOVE account, you can easily check what your school has access to. You can also watch the videos when you're off campus. And if you're an instructor, you can create embed codes to share the videos with off-campus students. You can use the account to create playlists of your favorite videos. And for instructors, you can also set up short multiple choice quizzes that go with the JOVE videos. Now, what is JOVE then in terms of products? So we create scientific videos for 5 resources at the moment, and these are aimed at helping with education, laboratory training, and research. On the education side, we have 3 products. First, we have JOVE Core, which is our series of animated video textbooks. Concepts in various disciplines such as biology and molecular biology through high-impact animations combined with real-life lab experimentation. So this is really aimed at helping students learn about quite difficult and advanced scientific concepts through these animations, and then they can see how real researchers are applying these concepts in their research in the lab. Then the next product is JOVE Lab Manual. And here we have sets of introductory lab experiments for biology labs and chemistry lab courses. Here, students can easily learn simple experiments that you can do with a group of your students in the lab. Each of the experiments comes with 3 videos: a concept video explaining the background of the experiment, an instructor preparation video that helps you as the instructor prepare the lab before the students come, and then there's a student protocol video that takes the students all the way through the experiment step by step. Then the next product, JOVE Science Education, is designed both for education as well as laboratory training. Here you find video demonstrations of how to use certain pieces of equipment in the lab safety videos, how to work with model organisms in the lab, and how to set up various experiments in over 8 different scientific disciplines. Research side of JOVE, we have more videos to help you learn the latest and most advanced techniques. So in JOVE's video journal, we publish method articles from authors all around the world. However, instead of just publishing the written version of the protocol, we also visit these people's labs and we film how they perform the experiment in their own laboratory setting. Then this how-to video, combined with the method article, will be published in JOVE's video journal. These video articles are fully peer-reviewed and are also indexed in databases such as PubMed and Web of Science. And then the latest addition to the JOVE family is JOVE's Encyclopedia of Experiments. And here you find collections of advanced experimental techniques, both in animated form as well as in lab demonstration form. And here we cover how to work with specific model organisms and how to set up cancer research models and cell biology techniques. So let's have a look at some of these products a little bit more in depth. So when you visit the JOVE website, you have a choice of visiting the education section or the research section. So let's first start in the education section. Here you can either search by topic or by JOVE product. So let's first have a quick look at JOVE Core. So JOVE Core is a series of video textbooks, and these can serve as a primary or a supplementary teaching resource. So these online video databases follow the same textbook structure as the most frequently used textbooks in the various fields. For example, JOVE Core Biology is based on the same chapter structure as Campbell's Biology. We have a JOVE Core Biology series. We have a chemistry textbook, organic chemistry, and molecular biology, as well as a social psychology textbook. In here, you find short animated videos that clarify specific concepts and scientists in action videos that show the students how these concepts are applied in a laboratory setting. Now, this is an example from one of our latest textbooks that we've just released, and it's on JOVE Core Molecular Biology. In here, you find a lot of information about how DNA, cells, and DNA replication works, as well as protein functions and the various signaling pathways that are very useful when you're studying molecular biology or when you're studying pharmaceutical sciences. In here, you find a number of chapters, and each chapter consists of several lessons as well as several scientists in action videos. The videos are nice and short and concise, so they don't take up too much of your lesson time, and are really helpful when you're learning these advanced research concepts. Then the next product I'd like to highlight is JOVE Science Education. And here is where we really start to learn about the practical aspects of working in the laboratory. We have 8 different series in science education, ranging from basic and advanced biology to chemistry, clinical skills, engineering, environmental sciences, physics, and psychology. Here we use a combination of animations to clarify certain concepts and then show students how to set up an experiment from start to finish, as well as give them an idea of what other applications these techniques might have outside this particular field. Now, on the science education videos, we have a lot of helpful features. So these are just a few to name for instructors. So these videos have navigation buttons. This makes it easy to jump to specific parts of the video. For example, if part of the experiment is a little bit more complicated, you can use these to navigate back to that part of the experiment. We also have the create a quiz option, and this is available on all of our educational videos. Here you can set up a short multiple choice quiz for your students to make sure that all the content is fully understood. These quizzes also give you a great insight to see where students are struggling to understand the matter, and so you can spend a bit more time during your lectures fixing those particular issues. Also, you have the embed video button, and this will help you get your videos into your course pages if you're using a learning management system. This is a very simple embed function, and we have detailed video instructions on our website, as well as a dedicated customer success team that can help you with embedding any videos you might like to use. We also have a detailed written protocol. So here's what the students can print and bring with them to the laboratory, and it has all the different steps detailed from the experiment. Some of our videos also have an interactive mode, and these videos can be found in our chemistry collection. Here, the quiz questions have been overlaid on top of the video to make the video interactive. It will pause and ask the viewer a question. You can select an answer, and when you get it right, continue watching the video. If you get it wrong, it will give you a hint, and you can select a different answer. Then moving over to JOVE Research. So this is where we find the more advanced research experiments. We have 13 different journal sections listed here, and each of these are fully peer-reviewed and fully indexed into different medical databases. So these you will find in JOVE Journal, and then we also have JOVE's Encyclopedia of Experiments. So JOVE Encyclopedia of Experiments is the latest addition to our family, and we have two sections at the moment, biology and cancer research. Biology details how to work with certain model organisms, and we've organized these by life stages of the organism. And in cancer research, you find cell biology and molecular biology techniques, as well as a lot of cell-based assays that are specific for the different types of cancer research, but are also very useful for anyone else working in cell biology and molecular biology. Now, in these experiments, you'll find the full experiment first in animated form, and this is incredibly helpful to understand the why behind each of the different steps. This will also help you when you're troubleshooting an experiment that just doesn't work, as it gives you great insight about why we take certain steps and why certain reagents are added. After that, the video jumps to an in-lab demonstration, and you can actually see someone perform this part of the experiment so that you have a great idea of what this will look like when you're doing it yourself. Also, these experiments all come with a detailed protocol, and this is something that you can bring with you into the laboratory. Then JOVE Journal. So this is our peer-reviewed scientific video journal, and it is the first of its kind. All of our videos are indexed in a variety of databases such as PubMed, Web of Science, SciFinder, and Scopus. And all of these come with detailed text protocols. So you get both the written version of the protocol, and at the same time, you can watch the video that shows you how the original authors performed the experiment in their own lab. You can also download a PDF of the article as well as a list of materials. And we also have citations that will help you use this article in your own research. We have 13 different journal sections, and many of them are very useful if you're working in pharmaceutical sciences and molecular biology. Then JOVE Journal has a publication process that is largely similar to how traditional publishing works. However, we have a couple of extra steps, of course, so I would like to take you through these in case you might be interested in publishing your own article. So our publication process starts by the submission of a manuscript, and this is in the form of a method paper. We do have a template on our website that potential authors can download and have a look at how the article should be structured. It should include a step-by-step detailed protocol and also a set of representative results. Our editors will look at your manuscript and then send it out for peer review. This works largely the same as in more traditional journals. So peer reviewers come back with suggestions, and when all that is resolved and the manuscript is accepted, we have our in-house PhD level script writers turn your manuscript into a filming script. So this is not something that you'll have to do yourself. Then when the script has been approved, we have a videographer that makes an appointment with your laboratory, and they film the entire experiment in one day. So this means we film the experiment kind of like you would a cooking show, and you need to have all the different steps of your experiments ready to go on that day. After that, we edit the footage, and we also add a professional voiceover that will narrate the entirety of your experiment. So this is also not something that you'll have to do yourself. When the video is done, we send you a copy of it so you can let us know if any work needs to be done further or if the article is ready to be published. When it's ready, the article and the video are published together on the JOVE website and are also indexed in the variety of different databases. Now, for some people, publishing their JOVE article with a video has helped them massively teach other people how to use this technique. So I'd like to highlight this example from the NHS Foundation in the United Kingdom, because they developed a new technique for shielding breast cancer patients' hearts from radiation. Now, they get a lot of training requests for this new technique, but they didn't really have the time to travel all over the world and teach people one by one how to use this technique. So they decided to create a JOVE video article and use that for training new staff. But also the training for other countries worked really well this way. So they found that they could train clinicians in over 40 countries in this technique, and 9 out of 10 requests for training were met by the article alone, which means no further explanation was necessary. So this meant they saved a lot of resources on travel and training, which they could reallocate into their research. Then how do you find things on the JOVE website? So if you're looking for a particular topic, what would be your best course of action? So if you visit jove.com and you've signed into your JOVE account, you can use the search bar to search for specific research protocols, model organisms, or any topics of your interest. By using the search bar, in this case, we've done a quick search for compound administration. It will bring you back relevant results, both from the JOVE Research side as well as the education side. So if you're training your students, you might want to start in expanding the research side, and we have various nice videos using a lot of animation to teach people how to administer compounds to rodents, for example. Now, if you're looking for a more advanced techniques, then you can look on the research side of JOVE. So these are articles in JOVE Journal and Encyclopedia. Now, if you found articles that you're interested in, and by the way, you can filter these results as well. So if you're looking for only articles in a specific subject area, such as biology or medicine, you can filter by that. You can also filter by institution, author, and publication date. Now, if you found articles that you find very relevant, a nice way to save these articles is by creating a JOVE playlist. Playlists can be used for teaching, they can be used for training your new lab staff, and they can also be used to collect all of the relevant protocols for your research. Now, playlists are shareable. You can decide whether or not you want to share your playlist with your students, your colleagues, or your lab staff. You can customize them, so you can make them look like what you need them to look like for specific training needs. And they're a great way to locate the videos that are of interest to you. They can also be helped with by engaging students more into the different topics by having all the videos in one place. Now, creating a playlist is super simple, but we've already got some playlists lined up for you on the JOVE website. So on the JOVE website, we have here the Faculty Resources Center, and it's got a lot of research and education playlists already ready-made for various scientific topics. For example, in our research section, so these are the research playlists, we have videos on lab training, we have analytical pharmacology lined up, we have stem cell facility, flow cytometry, and cell therapies. So there's a lot of content that is already lined up for you. Now, we also have some educational playlists already ready to go as well. So if you have a look in our chemistry section, we have a lot of playlists on, for example, chemistry labs, but also pharmaceutical sciences and an introduction to pharmacology. And these take students through the different steps. So there's nice introductions. There is introductions to kinetic pathways and to various drug kinetics as well. So if you want to know where to start on the JOVE website, having a look at the faculty resources page and searching out a couple of these playlists is a really nice way to get started. Now, this is an example of the faculty resources page, which can be found on the top of the JOVE website. So the first section is all the different playlists that we've created. And if you wanted to know how our videos relate to the different textbooks, we also have a section here called textbook mapping. So you can see exactly which textbooks match which JOVE videos. Also, below that, you'll find guides on how to integrate the JOVE videos into your online courses and your learning management system. So no matter which system you're currently using, we'll have a short tutorial video and the detailed steps of how to embed videos in there. If you're using the videos for streaming your lab courses or your lectures, for example, via Google or via Microsoft Teams or Zoom, we also have guides for teaching remotely using JOVE videos, so you can get the settings just right. We also have weekly training webinars that you can register and attend for free. These are demonstrations of the JOVE website, and you'll have every opportunity to ask any of your questions to one of our experts. We can help you with embedding questions. We can help you find content. So if you're interested in these, they take place on Wednesdays and Thursdays. So for you guys, the Wednesday session would be at 3 o'clock. Then if you are interested in having a playlist, but you're not quite sure where to start, or you don't have time to find content on the JOVE website, we have a team of experts who are running a project and training program mapping service. Here you can let us know which content you're interested in, for example, which classes you'll be teaching and what techniques you'll be covering. And our team will put together a playlist that best matches your course topics. So no matter if you're using this for training purposes, or you have to teach a class of first-year students, or you just want to know what content is available on the JOVE website that will be relevant for you, you can contact our team of experts and let them know which topics you're interested in, and we'd be happy to create a playlist for your specific topics. Then I've also put together some solutions for pharmaceutical sciences. So if you're on the JOVE website, you might wonder, oh, I don't see where the pharmacy section is. And that is because our pharmaceutical sciences are neatly divided between two of our very popular JOVE solutions. So we have our chemistry and environment content, which covers the chemistry aspect of it. It also covers some environmental sciences and biochemistry. Then if we're looking into the molecular biology aspect of pharmaceutical sciences, these you'll find in our biomedical sciences and clinical medicine content. So here you find everything biology and molecular biology related, as well as clinical skills, such as nursing skills, which have to do with preparing the solutions and also administering the compounds to patients. So by combining these two solutions, you have all of the content that is relevant for pharmacy and pharmaceutical sciences, as well as the molecular biology and medicine content. Now, finally, I'd like to conclude with some examples of some of the customers that are already making full use of JOVE's databases, including for drug development. So some of these you might recognize and some of these might be new to you, but these are all some of our happy customers that are already using the content. With that, I would like to thank you very much for having me here today. We are available for any questions. You can either or you can let us know via email. Thank you very much.
Thank you, Dr. Mikey Pauls. Thank you so much. So before proceeding further, I would like to welcome our guest of honor and our resource speaker, Dr. Mikey Gupta, sir. Sir, good afternoon. Good afternoon, sir. Good afternoon, sir. Dr. Mikey Gupta is a National Scientific Advisor of the Pharmacovigilance Programs from India. He is President AIMS Bhopal and President AIMS Jammu. He is a Principal Advisor THSTI DBTI Government of India and Chairman of multiple government and international science bodies throughout this world. We welcome you, sir, on this one-day international seminar on Advancement in Pharmaceutical Education and Research, Current Insights into Molecular Biology Techniques. Welcome you, sir.
Thank you very much, Dr. Ram. And I was carefully listening to the talk of Dr. Pauls. And my bad luck, because I have this simultaneously another VC where I had to deliver a talk, so I could not concentrate it. But I will just comment on few things because I have gone through this your video journals. It's one of the very exciting area, I must say, and that is what is required for today, not for publishing, only for getting the impact factor and citation, but more important thing is, if you have developed any technique, if you have done anything, then how the world can replicate it, how the world can understand it. And in that, I have seen this video some time back at I do not know which place, and I was so much impressed that this is the way how what innovation one is doing in the lab can be translated, can be trained to others. We do a lot of, Mehan will agree with me, and I have requested all the scientists in Moga college and the College of Pharmacy,
Yes, sir.
to organize a hands-on workshop. See, if I say that their scientists in Moga are doing so critical experiments in their lab and so much innovation which they have done, and what they are doing or what we are doing, we are just publishing in a very good impact factor journal. But the real benefit will come when people will see you doing through the journal, which is a video journal, where it can focus very accurately the tricky points. That will be really useful, and this will not only be useful for researchers, but this will also be useful for M.Pharma PhD students who want to learn techniques. And I would also maybe Paul's make later on clarify sometime that will that amount to patenting something like that, because once you have done it, you have shown it, if some person does it again with the same technique, same demonstration, how that would be protected? Will that be called as a plagiarism, called as patent protection? Because if I develop a new technique, maybe slight new improvement, which will cause more accurate localization of to the brain area, or maybe destruction of some neurons, like that. So that is one thing. But I must say that you have chosen by default or by chance wonderful institution which is doing extraordinary innovative research and publishing paper nicely. I can see Dr. Shyam Sundar Sharma from Naipur, Mohali here, who has also done tremendous innovative experimental model on diabetes and stroke, which I think can make a lot of difference. So I congratulate Dr. Paul for your journal, and I'm sure that this is the need of the hour. I would also congratulate, namaskar, and I would also congratulate the College of Pharmacy, Dr. G.D. Gupta, who always is so enthusiastic person. I think you should also take some DNA from his sample and identify and what is the reason how he gets so much of energy and to mobilize so effectively so many things. So congratulations and the leadership of ISF College Moga. Now coming to just 2-3 minutes, because on the molecular or the sophisticated techniques in pharmacology or say biomedical sciences, the instrumentation, documentation, validation, and reproducibility is the most important factors in science today. If we are not having adequate infrastructure, adequate instrumentation, adequate facility which can address our question, our hypothesis will not be able to be, we are not able to prove it. So I always say that first thing is you have to generate an hypothesis. The second thing that you have to generate the adequate and appropriate tool to prove that hypothesis. If you do not have appropriate tool, you will not be able to prove it. And these tools would be either a statistical tool if you are doing tabletop exercise, or it can be instrumentation, or it can be the survey type of thing. When you talk of an instrumentation, then not only the instrumentation which go into the depth of the problem, that is you do the molecular biology, and that is not only useful in research, but which is also useful in a translational work. Now you just see the corona, it is the classical example of a molecular or high-tech research. We used to talk that vaccine development takes 15 years time and $1 billion rupee or $1 billion. Now it has reduced to less than 3 years, and the vaccine has been developed because the techniques. High throughput screening, now nobody does the conventionals. You test with the high throughput, and hundreds and thousands of herbal compounds, or maybe whatever the in vitro, in situ compounds you're doing, you can funnel it into 100 or maybe less than 100 in a very short time. It reduces cost, it reduces time, and it reduces the chance of failure and improves the chance of success. So the important thing is how to validate these processes. The validation of any process requires that this process must be done at more than one place. And then when you validate it, it has to have reproducibility. Whether it is a 90% acceptable reproducibility or 95% or 92%, that will be decided based on the power of the study. And that is what one has to decide on the validation. And when you do validation, it across the lab difference in the value which you get it, because in the molecular biology or in the micro instrumentation technique, the lab to lab variation will create havoc. And that's why when you do standardization, not across the laboratory but across the trans-border boundary laboratory accreditation is. And that's why you see many clinical trials, we have a centralized laboratory. Why we have to have a centralized laboratory? That means we believe that the laboratory in Moga or laboratory in Delhi or laboratory in Geneva will have a different results. No. The idea is that these validations should be such that the data or the value which you produce from one lab should be acceptable to other. And that's what the importance of the GLP, good laboratory practices here. I chair that committee, and Dr. Shyam Sundar Sharma is a member of technical committee, and we have now just in 2 days back, we have decided that the next workshop on GLP will be conducted by Department of Science Technology in ISF College of Moga. So this is news I wanted to give, but I thought that I will give it during this occasion. And so there will be next meeting, next funded program by Department of Biotechnology on GLP will be done for that. So with this, I stop here, congratulate all the faculty members, and I really appreciate the journal, and I wish that you make this journal at least for few years very subsidized or low cost, so that the people are hooked to it. Once the scientists in India get hooked to it, I think you will recover whatever the free ship you have given in the initial year. Thank you, Pauls, and thank you, Dr. GD, and thank you, Dr. Shyam, and thank you, everybody.
Thank you, sir. Thank you so much for your inspirational words and your valuable information on this platform. Mr. G.D. Gupta, sir, so should we proceed further, sir? Okay.
Continue.
Okay, thank you, sir. So, dear audience and respected speakers, guest of honor and speaker, Dr. Shyam Sundar Sharma. Dr. S.S. Sharma is a professor in the Department of Pharmacology and Toxicology at the National Institute of Pharmaceutical Education Research, Naipur, Mohali, Punjab, since 2009. He worked as a postdoc fellow at the University of Illinois, Chicago, USA, and he did his PhD pharmacology at All India Institute of Medical Sciences, New Delhi. Dr. Sharma has published more than 150 research and review papers, patent books with more than 5,000 citations with a valuable H index 40 and I-10 97. Dr. Sharma research interest includes understanding the potential role of pharmacological agents in CNS disorders, diabetic complications, cardiovascular disease, and safety pharmacology. He has more than 20 years of teaching and research experience and guided more than 100 masters and doctoral students. We welcome you, Dr. S.S. Sharma, on this one-day international seminar. Sir, please proceed.
Okay. Yeah, thank you. Actually, first of all, I would like to thank Professor G.D. Gupta for inviting me for this very nice symposium talking about the advancement in the molecular biology techniques in pharmaceutical sciences. And it's really great honor to have Professor Y.K. Gupta as one of the chairman of.
While I congratulate Ashraf and their entire team for wonderful imagination as
Yes,
Okay.
Dr. Sharma, you continue.
Yeah,
Please.
I think yes. Okay. So that way, I think I thanks Professor D.D. Gupta for inviting me over here actually for this talk. And I thought like because since students are joined in this particular group, so I thought like just talk about some of the molecular biology techniques which can be used in pharmaceutical sciences. Though we are not using all of them actually in NIPER, but like definitely throughout India and throughout the world, many pharmaceutical scientists they would be using this. As it was mentioned that I think I was also impressed with the JOVE because they are creating a lot of new experimentation and many times we get a free trial. Recently, we also have got a NIPER has got a free trial for GOVE, actually JOVE. So that's I think all the NIPERs are going to be going to enjoy for some time free trial. Earlier also we have seen many videos actually on JOVE. In fact, actually, like when I was at Ames, we used to work on the stroke model, middle cerebral artery occlusion induced model. At that time, actually, just creating that model and all, actually, sometimes we have seen the videos actually. And even like for some of the stereotaxic techniques and all, we do it continuously in our lab. We also like very familiar with those. And sometimes whenever we need and we get some free subscription, then only we are looking at it. But now I think NIPER consortium might plan to get a subscription for JOVE actually in future because I think free trial might create a opportunity for all the scientists, students to learn about JOVE and share their experience on that base. Like NIPER can definitely have subscription actually from the JOVE. Okay, so let me. Yes, let me share my. Well, like my presentation is visible.
Yes, sir.
Okay. So I think like the way actually like I
Sir,
thought how
sir,
this yeah
sir, sorry to interrupt. I request
yeah
to just zoom it, sir. Zoom it.
I
Slightly.
have zoomed it actually I think
Sir, sir, right side below corner, if you can zoom it, so that slide will be more.
okay. Can you see that one or no?
No, sir. Sir, you just slide share. I think not visible. So you can from the zoom ratio 70% to 90%.
Okay.
Yes.
Okay. Yeah, it's there. I think this is a problem with the. uh,
Now it's good, sir. Now
yeah,
it's
because like this is a problem coming with the Google Meet.
yes, yes. Now it is visible, sir.
Okay. Okay, in this actually, like just I thought some of the points already mentioned by Professor Y.K. Gupta that I think in India, whenever anybody is doing research, one of the thing actually like which need to be taken into consideration the reproducibility, and always like I think last time when Professor G.D. Gupta organized one talk on research integrity, At that time, I talked about the ways actually how you can improve and how we can train our scientists so that research integrity or research ethics need to be maintained. And why? Because most of the time, whenever we are going to use these molecular biology techniques, I think everyone must write standard operating procedure (SOP). And in GLP, good laboratory practices, I think first thing is that for each and every activity, even the equipment, nowadays we are getting a lot of equipment from abroad, and now under Make in India program, now the government want some of those instrumentation can be developed in India itself actually. So that's why in future I think calibrations of these equipments also going to be one of the issue, and we have to have a compete with the world for if we are going to make these kind of equipments which are being used or which are being used for molecular biology techniques. I think the techniques most of you would be knowing, but just for the students' point of view, I would like to mention that molecular biology techniques are common methods which are generally involve manipulation and analysis of DNA, RNA, protein, and the lipids. So now, all the techniques can be used. Actually, several techniques have been already developed actually, which are being used, and in future we can see a lot of other techniques. And these techniques are not only used in case of biology; they are also used in chemistry, physics, and now in pharmaceutical sciences also. Some of the techniques which have been mentioned over here, DNA gel electrophoresis, Western blotting, and various blotting, PCR, polymerase chain reaction, next generation sequences, arrays like microarrays, cytokine array, protein arrays, protein ligation assay, knockout, knockdown strategies, they are being used. And some of the fluorescent technique like FISH, ELISA, immunocoprecipitations, immunohistochemistry, immunofluorescence. And out of that, mostly some of them actually even techniques to confirm localization of some of those protein like IHC, immunohistochemistry, immunofluorescence, and they also have been listed over here. And when it comes to pharmaceutical sciences, now you see that most of the biopharmaceuticals, they are involving the development of the biopharmaceuticals actually, whether you take development some of the monoclonal antibodies or polyclonal antibodies actually, they involve a lot of molecular biology techniques like expression of different enzymes, proteins, and all actually, and then getting those pharmaceuticals. In drug discovery and development, this target identification, target validation, pharmacodynamic biomarker monitoring going to require the use of molecular biology technique. And in target identification, if the target is the nucleotide, then DNA microarray cloning can be used, sequencing can be used. In protein level, like Western blotting, or protein arrays like protein array, proteomes, genome, like all genomic proteomics, actually, those kind of things actually, which are being used for these kind of investigations. And even target overexpression, which is being used for transfection studies. And what has been seen that this target down regulation like RNA interference, siRNA, shRNA, knockout animals, they are being used for various investigations. So that is a significant number of molecular biology techniques are being used. For pharmacodynamic biomarker monitoring at the nucleotide level, like FISH, fluorescence in situ hybridization, northern blotting, real-time PCR, protein level, immunohistochemistry, Western blotting, then this facts like fluorescence activated cell sorting, enzyme link, immunosorbent assay like ELISA, they are continuously being used. If we come to this, one of the commonly used technique for detection of the end product of the PCR, like DNA gel electrophoresis, this is a way you can see that DNA samples are being loaded on the well, and then you can see that there's a separation based on the charge, from a negative to positive charge. Actually, you can see there's a separation on the gel actually, and then you can identify the largest and the smallest, like these kind of separation. And using these particular equipment actually, which is connected with the power supply, you can do it actually. The one of the video actually, like you just see that how this can be used
for separation of DNA, RNA, and sometimes large proteins. It is widely used for the separation of DNA. Depending on the size of DNA which is to be separated, concentration of agarose is kept between 0.8 to 2%. Agarose is prepared by dissolving agarose in an appropriate buffer. Usually TAE or TBE buffers are used. The same buffer is also used in the buffer tank to run the gel. The mixture of buffer and agarose is boiled for a few minutes. The boiling ensures that the agarose is uniformly dissolved in the buffer. The agarose is then mixed with ETBR and poured in the tray. Once solidified, the gel can be used for electrophoresis. For analyzing DNA samples, about 2 to 5 microliters of sample is mixed with a drop of bromophenol blue and loaded in the gel. Bromophenol blue is a dye used to monitor electrophoresis. Electrophoresis is carried out at an electric field of 5 to 10 volts per centimeter. Remember, electric field is the applied voltage divided by the distance between the electrodes. The direction in which the DNA runs is from negative to positive. Once the run is complete, the presence of DNA can be visualized under UV transilluminator.
So this is the commonly used techniques actually, which most of the labs would be using, and I've seen some of the papers from ISF Moga about like using the protein for protein like Western blotting, which is being used, and generally like you know that protein separations actually can be done on the gel actually, like, and then transfer is done using the transfer apparatus, and using then you can identify those protein using primary antibodies actually, and then identifications actually, this quantification can be done once you have identified those protein, and then quantification can be done using gel doc system. So we use actually like this Bio-Rad gel doc system for these kind of studies. One of the experimentations I just would like to see that what we have done in our lab, where we showed that effect of TRP channels inhibitors in the model of Alzheimer's disease. I would like to tell you that I think all of you will also be remember that recently in 2021, October 2021, Nobel Prize was given to the work related with the receptor through the touch and the temperature, and that is TRP, transient receptor potential channel. David Julius and the Ardem Patapochian, they were awarded actually this prize, Nobel Prize in Physiology or Medicine, for their discovery of receptor for temperature and touch. And as they were the one actually discovered this TRP, and we were fortunate enough that we are working in this area, and we have used some of these TRP channel inhibitors in the model of Alzheimer's disease. So these Western blotting studies which we have done actually, like you can see there, this is the gel actually control diabetic plus the treatment with trenilast actually, and you can see there like the drug beta amyloid model. You can see over in the next panel, a sham operated animal and beta amyloid. In these studies, beta amyloid was administered by ICV, intracerebral ventricular cannulation, directly to the brain, and then we have checked the cognitive functions, and there was significant impairment with the cognitive function for observed. And then we use this TRP channel inhibitor trenilast at the dose of 30 and 100 mg per kg, and then this quantification is being done in two models, like diabetes-induced cognitive impairment as well as the beta amyloid-induced cognitive impairment. And we found like trenilast was significantly reduce the expression of TRPV2. So and this Western blotting technique can be used in various investigations, in diagnosis also, and just to find out the effect of any interventions on some proteins, actually, whether those kind of like ion channels or receptors or anything you can do it. Unlike blotting technique, which is generally used in pharmaceutical sciences, also the northern blotting, particularly for detection of this RNA. You can see, you can take the sample, then RNA extraction is done, and then again, similarly, like gel electrophoresis is done. Then RNA is fixed to the membrane with the UV or heat. And then you can visualize using labeling or on X-ray film. You can go for quantification, and these techniques are used for various gene expressions to observe the overexpression of the cancer-causing gene and the gene expression in case of transplant rejections, Crohn's disease, and many other cases. Then southern blotting also used for this genomic DNA, like you have seen the northern blotting that was used for RNA. This is used for DNA identification, and that detection is like you can see the probe. This radiolabeled probe can be used for finding out the particular DNA sequence. The detection of the mutation, deletion, or gene arrangement can be done using southern blotting. So that's also technique is commonly used in many labs, actually, particularly in many medical institutions that is being used. And in addition to these techniques, molecular biology technique, there are techniques which can also be used for identification of the protein, like immunohistochemistry or immunocytochemistry can be used, where like in the live cells, you can identify, or maybe in the fixed tissue, you can identify the protein. And I've seen like many papers from ISF Moga, actually, like many of the faculty, they are doing immunohistochemistry or immunocytochemistry experimentations. And what you require, like for this, you should have a fluorescence microscope in your lab, actually. Okay, and other techniques, actually, it's like primary antibodies and secondary antibodies, and then you can identify those kind of proteins. Like we have done some of the experimentation related with the immunohistochemistry. You can see that in control animal and the diabetic animal, where we have just looked at the Wnt signaling pathway. There you can see the diabetic animal, they did expression of the Wnt protein, actually, and then whether the inhibitors of like this Wnt signaling pathway like LGK can reduce this immunohistochemistry. Even immunofluorescence techniques is being used, actually, where you can identify using again antibodies, where you can see that fluorescence has been detected in some of the samples of the paw, actually, where we have done this related with the nerve conduction velocity. Actually, some of these experimentation has been done. Polymerase chain reaction like PCR is being used, actually, like whenever you have a small sample, actually you can amplify the DNA, and which can be used for further experimentations, actually. And you can see that you can check using the PCR cycle, actually, like you can increase the amount of DNA which can be used for further analysis. And these kind of the bands, actually, like you can get whenever you run these kind of gel, actually, like these kind of band can be there. One of the technique which have been commonly used, I think everyone have seen that this COVID-19 reports, and in that confirmatory test for this COVID-19 SARS-CoV-2 was done with real-time PCR, and it can be both qualitative as well as quantitative technique, and this quantitative PCR is one of the most used technique for mRNA quantification in translational study. So in this study, like this sample are taken, and then this RNA is isolated, and which is converted to this complementary DNA, and then it's a quantitative PCR is run, and then you see the quantifications of this technique. Even the quality of the RNA can be checked, whether RNA assessment can be done, and then using reverse transcriptase, actually the cDNA convert to again to RNA, and that can be looked into various changes on gene expression. RT-PCR has been used for disease diagnosis, cancer genetic, infectious studies, SNP analysis, like single nucleotide polymorphism analysis, that can be seen actually in different patient-to-patient variations actually of drug response as well as the disease, that you can also do using this one. Some of gene expression studies we have done in our experimentations on this amyloid model. What you can see that in sham operated animal and in amyloid animals, actually you can see there is a significant increase in the gene expression, TRPV2 gene expression. And this TRPV2 gene expression was significantly reduced by this trenilast 30 and 100 ng, which is a TRPV2 inhibitor. And then TRPM2, so we did actually various like TRPV2, TRPM2, and we looked into whether they are involved in. So this is the one way that to find out the target, whether this TRP can be the target for Alzheimer's disease or not. So these kind of experimentation can be done. Co-immunoprecipitations can be used actually by many scientists, where just two protein, whether they are interacting with each other actually. So that can be also done actually, these things actually. So these kind of techniques are being used. And this chip assay is also commonly used actually for identification of some of the link actually between the genome and the proteome actually, by monitoring the this transcriptional regulation through histone modification. So for epigenetic studies, this chromatin immunoprecipitation assay, some of the faculty in NIPER, they use this chip assays for epigenetic modification in diabetic cases, cancer cases, and all actually that is being used actually. Proximity ligation assay for protein identification, that's also being used actually, this one. In addition, that even this DNA fragmentation, this apoptotic pathway, where the death signal, particularly they act on, we call FAS, first apoptosis signal, FAS. FAS act on the FAS ligand, act on this death receptor, and then initiate the series of events leading to apoptotic cell death. And apoptotic cell death involves the DNA fragmentation. So now these apoptotic detection assays, you can see there is a list of apoptotic detection assays actually, which can be used actually. So they're morphologically you can do using electron microscope. I think NIPER Mohali has electron microscope that has been used for identification of the apoptosis. Genome fragmentation can be done like DNA layering. Actually, we initially told about the DNA and gel electrophoresis. Tunnel assay can be done, and some kind of annexine or caspase, and those can be done. But generally, this tunnel assay is being used, which is basically terminal deoxynucleotide transferase mediated DUTP nick and labeling assay. So in this, wherever cells are there's a apoptotic damage, this green fluorescence would be shown. Wherever you can see the green fluorescence means that there is a apoptotic death. So in case of stroke animals, you can see in the this one normal sham and the normal ischemic reperfused animal. This white portion or the pale yellow portion showing there is ischemic damage. So when we have subjected this damaged portion for apoptotic damage, we found there is significant increase in the apoptosis with that. One of my PhD student, he investigated the effect of 3-BNI, 3-bromonitroindazole, which is a NNOS, neural nitric oxide synthase inhibitor, on the DNA fragmentation, and showed there was a significant reduction in the DNA fragmentation with this particular treatment. ELISA is commonly used by every lab. I think this is very common technique nowadays being used to check this TNF-alpha, interleukin-6 levels. Kits are available for rat, human, and all. Actually, you can use those kits actually. But because of this pandemic, now these kits have become very costly actually to afford, but earlier they used to be much cheaper actually. So this TNF alpha, there is significant increase in the diabetic animal, which was decreased by the treatment with one of the antioxidants, sulforaphane, actually, like you can see. So these kind of experimentations can be done. And this very common technique, which is being used for in pharmaceutical sciences for various history. DNA microarray can be used to assess the gene expression, like global gene expression, like proteomics. You can use like protein array, like for gene expression, you can use DNA microarray, and this can give the idea about the whether there is a gene upregulation or the gene downregulation. So at a time, you can measure like many genes actually in one go, and you can compare with the normal with the disease conditions, and that can be done actually. So there are like various DNA microarray are available for apoptotic gene, oxidative stress gene, and various other genes actually you can purchase, and you can do this microarray. One of the study in which were published in the PLOS one, they did microarray in Parkinson disease research actually, and they identify some of the microarray which was changed in this mouse model. Cytokine array can be done nowadays. Actually, you can identify the number of cytokines. What are the different cytokines which are altered by the treatment, okay, and which are altered in case of disease. Those can also be investigated. Protein array can be done nowadays. Actually, like when we have the gene expression, like we have about like 30,000 plus gene, you can analyze in one go. Similarly, protein array can be done, like 2D gel electrophoresis can be run, and when you get that one, actually you cut those blots actually, and then analyze using mass spectroscopy like multi-top, you can find the details of those molecular weight of those protein. The protein array can be done, and that can be that have been used for various identification of the alteration of various protein in cancer patients also. And you have a sample biopsy from healthy and the breast cancer biopsy, and then you'll identify that. And these kind of like investigations have been used even for precision medicine. So in pharmaceutical sciences, another area which have come up with the precision medicine, and these techniques are being used. Fluorescence in situ hybridization, like fish techniques, can be used, and nowadays most of the labs, like they have those confocal microscopes and all actually, and they can identify some of these changes, and fluorescence in situ done even. Next generation sequencing can be done to identify the sequence of and gene annotation, SNP, and all like changes or different like comparison of the different two individuals that you can identify. Okay, these are the individual are having single nucleotide polymorphism actually. So there might be the variation in the effect of the drug, like pharmacodynamic variations can be there in those cases actually. Whole genome and the transcriptome profiling have come up actually. In India also many labs have started doing this whole genome and transcriptome profiling. Even there are many facilities are available throughout India. Actually, you just submit your sample and they are going to provide you the this sequencing of your sample. So the sequencing for epigenetic studies, expression analysis, non-invasive prenatal testing, all can be like chromosomal like testing that can be used, like they can use sequencing method. Another very good molecular biology techniques have been used for overexpression, like target overexpression, like you know that in hypertension there is a increase in the like angiotensinogen gene expression. You can assess, okay, if whether the patient is having increase in the expression of the renin, increase in the expression of angiotensinogen, or what, depending on that one. And then you can find out, okay, this is the one, actually, this is because of overexpression of that particular gene actually. So those kind of like studies can be done. Some of the cells actually which do not have those kind of receptor, like we use generally for hERG assay, one of the assays which being used for detection of the QT prolongation or potassium channel blockade. These CHO cells, actually Chinese hamster ovary cell, they can be transfected with the hERG channel to look into like whether potassium channel expression is there. Then you treat that with various new chemical entity found whether there is a blockade of uh, that channel, actually potassium channel, using patch clamp. So we also do, actually one of our scientists is doing patch clamp assays for that. And this target validation method, like another like silencing, like gene silencing, that can be used, like RNA interference assay can be done by knockout animals. These investigations are being used for pharmaceutical sciences. Expression cloning, actually like that is being done for production of various pharmaceuticals, actually to increase the productions of various like uh, proteins, actually that can be, like suppose like you are looking into for interferon gamma, actually, and the yield of the current assay is less. You can have the technique, you come out with novel method, you can increase the expression of those TNF alpha, actually the yield will increase, and you can have the good, if you have a good yield, then you can definitely convert into the good product and can sell it. Another like common, or maybe I can see in the last decade, actually these techniques have come up, actually, and will find a role in pharmaceutical sciences. This CRISPR-Cas, this is nothing but clustered regularly interspace short palindromic repeats. And in this particular techniques, even you can do, like using this technique, you can do the gene editing. So this 2020 Nobel Prize in chemistry gone to Emmanuel Charpentier and Jennifer Doudna for discovering this CRISPR technology, and that was being used for drug discovery. So these techniques can play important role in future in pharmaceuticals and drug discovery, and you can identify the gene whether they're responsible for the drug resistance and the gene which causes genetic disease. So this kind of gene editing is being used as one of the technique, actually. Just see this video.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes into the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop code on a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A crab domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has faced, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
So this is actually, I think, gene editing technology in future going to be used for all kind of gene therapy. You know that one of the costliest gene therapy is 16 crore rupees for like one time gene therapy, which come up for treatment of spinal muscular atrophy. Knockout animals which are being used for pharmacology research throughout the world, and you can see that there are several of those gene expression. If you increase angiotensin receptor, if you increase the gene for like gene expression of angiotensinogen, renin, they can increase blood pressure. If you increase the expression of AT2 receptor, angiotensin 2 receptor, converting enzyme, if you decrease the expression of angiotensinogen, that can decrease blood pressure. So now, like just understanding a pathophysiology of hypertension, you can use a knockout or this knock-in animals cycle. So there are several like examples which are already done, like transgenic mouse model of hypertension. They are basically like using kind of an overexpression of like these protein like angiotensinogen and then this renin and all. Actually, like they have been used by many investigators. Like gene knockout also have been done. If you do the gene knockout for AT2 receptors, endothelin knockout actually found to be lethal. They can angiotensinogen knockout. So these animals will show decrease in the blood pressure. Pharmacodynamic drug monitoring can be done at the like nucleotide level to identify like whether like drug molecules are altering the RNA, or maybe like using this phage fluorescent in situ hybridization, they can identify whether they are altered or not. So now I think like many molecular biology technique and other techniques that have been used, actually, they play a very crucial role in answering the several impending questions in the field of pharmaceutical sciences. And these techniques are useful in design of diagnostics and therapeutics, which could be useful for the mankind. And molecular biology techniques already playing significant role in the field of biopharmaceuticals. Production of the many facilities, actually, like that is why you require a cell culture facility. Many of our discipline in pharmaceutical sciences, particularly like pharmaceutical biotechnology. If like in pharmacy institution we develop this discipline of pharmaceutical biotechnology, definitely we can contribute significantly to the area of biopharmaceuticals, actually, for developing newer technologies for various monoclonal antibodies, various protein generations and all. And you would have seen that recombinant DNA technology already being used for streptokinase like like recombinant plasminogen activator, and then more than actually like different like 500 monoclonal antibody protein therapeutics are available in the market, and future is going to belongs to the biopharmaceuticals, and the molecular biology field have potential to revolutionize the discovery in future. With this, I would like to thanks like my students because they were the source of inspirations always, and this is the campus of NIPER Mohali, and I invite you whenever you're coming to NIPER Mohali. Actually, you are welcome to visit.
Thank you. Thank you, Dr. S.S. Sharma, sir. Thank you so much for this highly informative and the scientific information regarding molecular biology techniques, their therapeutic values, their pharmaceutical significance in biomedical sciences. And sir, really, it's such a very wonderful presentation, sir. We see after a very long time, and we are very thankful to you to deliver this speech on this platform, the ISF platform, sir. Thank you so much, sir.
Thank you, Siddharth. Thank you, Siddharth.
So I request to all dignitaries, speakers, and guests, please put your face on so that we can interact and we can conclude the session in a good manner. I request to all members who join now to please put on your videos. Thank you. I want to take a very simple question from Dr. Mike Pauls. So, yes, Dr. Mike, am I audible to you?
Yes, you are.
Yes, okay. So, Dr. Mike, how these techniques can be beneficial in pharmaceutical education and research?
Okay. So in terms of education, we developed the videos so that students get the full package. So we start by explaining the concepts behind the techniques. How does DNA replication work, or how are proteins made, or what are proteins even? From education and teaching people the basics to inspire people to develop their own advanced techniques, which they can then also publish and share with the world.
Thank you, Dr. Mike, for your kind words regarding the utilization and the beneficial of these techniques. So I request, and I take another question to Dr. S.S. Sharma, sir. Sir, there is a basic question that how these techniques like PCR, Western blot, ELISA can be useful and be beneficial in especially pharmaceutical research institutes and the students who are pursuing M.Pharm, master degree or PhD in life sciences.
I think like the techniques are most of the time like somebody would like to work do the research, then definitely like I think they can use, like we are using actually ELISA every day. We are using Western blotting technique for, as I showed some of the results related to my TRPC channels, and we also doing like kind of a gene expression studies. So they are like commonly used techniques which I think most of the institution would be using. Still like some of the technique like gene sequencing or CRISPR and all, actually there are a few institutions actually in the Tri-City, they also using actually these techniques. So these techniques are not new actually to most of us, but like depending on the need actually. I think when you're talking about pharmaceutical sciences, we always think about related with the drug, like whether the drug is altering DNA, RNA, or protein. So our focus is like what is the effect of the drug actually, if any drug, how is going to alter RNA, DNA, and the protein. So always our focus. In pharmaceutical sciences, we can definitely use most of these techniques, and master students actually and PhD students, they must be exposed to these techniques. And education terms of I think like nowadays instruments are not that costly. You can also expose to B Pharm students, even the bachelor students also regarding these techniques, so that they will feel confident actually. And in case if you don't have the instrument, then I think you can take help of JOVE. Actually, they would be having videos, so they can see the video at least. Okay, and that can also help actually.
Thank,
So I think like JOVE actually like one thing I can tell, because we do a lot of pharmacology experimentations, and some of the experimentation, like one of the another company AD Instruments, they also develop some software for students for doing actually like teaching purpose. So I think like if you can also like see that, because I didn't because that is very basic level software they develop. So I think if the video like you are developing, if you can tie up with that, so you can also develop videos, experimentation videos for pharmacology students. And pharmacology subject is not only there in pharmaceutical science institution; it is one of the core subject in even in the medical school. So that way, I think you will have a lot of sale of these kind of product actually, if you can develop in collaboration with some of those physiology equipment companies.
thank you, sir.
Yeah, I agree with that. So we already have some of those collaborations, but definitely it would be great if we can do even more of those in the future.
Yeah.
We are also developing a lot of new videos every year. So I know pharmaceutical sciences and it's definitely high on our list of things that we want to produce in the future. So do keep an eye out.
Yeah, great.
Thank you, Dr. Mikey Pauls and Dr. S.S. Sharma. I want to share a very small experience with my experience with JOVE. Sir, last year we were planning to isolate the cerebrospinal fluid from rat spinal cord. So at that time, we were very challenging these procedures regarding how to isolate the cerebrospinal fluid, especially from the rats. Then a video which was came in my computer in front of my screen. One of my students gave me that video. Sir, this is a video which is present on JOVE. So I request to JOVE. They give me a 1 week subscription, and that was a tremendous time for me to how to isolate the cerebrospinal fluid from the rat cisterna magna. And finally, we successfully proceed this procedure. And recently, last week, we published a paper in Phytomedicine, and where we mentioned that method, that how we collect the cerebrospinal fluid. And I think this is very beneficial for all researchers and the persons who are in the molecular and biomedical fields. So now I would like to invite Dr. G.D. Gupta, our respected director, to please enlighten this lecture. Sir, Dr. G.D. Gupta, sir. I think. Okay. So I would like to invite our one of guest of honor and Dr. Dharminder Singh Gosai from JOVE India. So for vote of thanks. Sir, please proceed.
Thank you, Dr. May. Thank you, Dr. May. Honorable dignitaries, distinguished guests, respected faculties of ISF College, my colleagues from JOVE and all the participants. First of all, a very good evening to all of you. On behalf of JOVE, ISF College and the entire organizing committee of this webinar, it's a matter of pride for me to propose a word of thanks. First of all, my sincere thanks to Almighty God. Now I'd like to extend my hearty thanks to Sri Praveen Garg, Chairman ISF College of Pharmacy, for encouraging us to have this kind of event. Thank you, sir, for giving us an opportunity to organize this event. I would also like to thank Mr. Janesh Garg, Secretary ISFCP TLF, a global school in Moga, for his support in this event. I would also like to thank or take this opportunity to thank Professor G.D. Gupta, Director, ISF College of Pharmacy, for being a guiding force for this event, without which I don't think this event was possible. Thank you very much, sirs, for giving us this opportunity. We also look forward to your support in making our association, I think, more stronger with the time. Let me also express my gratitude to Professor R.K. Narang for his continuous support and guidance in organizing this webinar. Thank you very much, sir. I want to express my sincere gratitude to our chief guest for this event today, Dr. Y.K. Gupta, President AIIMS Bhopal and AIIMS Jammu, who gave his precious time from his busy schedule to grace this occasion. Thank you very much, sir, for your time and welcome address. We all are inspired by your great words. I would especially like to thank you for your for putting emphasis on the real benefit of JOVE and acknowledging its contribution in the reproducibility of the research work. To Professor S.S. Sharma, Department of Pharmacology, Naipur, Mohali, for his talk today. Sir, thank you very much for your excellent detail-oriented presentation on molecular biology techniques. We are grateful for your time and effort you to share your thoughts and experience with us. I would also like to express my sincere thanks to my colleague, Dr. Micah Pauls, for a wonderful presentation today and walking us through the JOVE platform, showing how faculties and students can use this video in their day-to-day teaching and research activity. Mike, I'm sure you're going to get many questions over the email regarding how they can actually use these techniques in their day-to-day activities. And how can I forget to thanks Dr. Siddharth Mehan, who is the force behind this event right from the beginning, when I discussed the idea of actually having a webinar with him. We actually discussed this webinar of having with only ISF College, but then he was very much instrumental in making it as an international seminar. Thank you so much, Dr. Mehan. Without your contribution, this event might not have been possible.
Sir,
so thank you so much for your such generous contribution.
I want to clap for Dr. S.S. Sharma for wonderful lecture. Sir, we are very thankful.
Yes,
definitely,
yes.
Sir. Sir, your session was very informative. We are having lot of congratulations regarding sessions or YouTube live parallelly we are operating, and excellent response, sir. We are very thankful to you.
Thank you.
Thank you so much, sir. And my deep sense of thanks and appreciation go to all the participants who are live with us today on YouTube and attended this event with the great enthusiasm. I think we had more than 200 people live with us on YouTube, and maybe a lot of people who are coming and going. I'm sure you all will understand that the event like this cannot happen overnight. The wheel starts weeks ago. It requires planning and a kind of bird's eye for detail. We have been fortunate enough to backed by a team of very motivated and dedicated colleagues from ISF College, and also the team of marketing from JOVE. We are very thankful to them for their support in making this event successful. Thank you one and all, and have a pleasant evening.
Thank you.
Okay. Thank you, Siddharth.
Thank you, sir. Thank you so much.
Nice meeting, Dr. Dharmendra. Nice meeting, Paul.
Thank you.
Thank you, Dr. Pauls. So thank you so much for your great presence and to make this seminar in a very wonderful and scientific manner. Thank you so much. Thank you, sir.
Thank you, everyone. Thank you so much. Thank you.
So I request to dignitaries, you can left the group so we can end this session. Thank you, Dr. Maike, Dr. Dharmendra, Kamal Singh, Bindia, Momita, and Randhir. Thank you so much. Take care. God bless you.
Thank you, everyone.
Thank you.